I'm Jared Zystro, and in my day job I work for Organic Seed Alliance, but for the past several years I've had the pleasure of working on a PhD under Dr. Bill Tracy, and so I'm going to be presenting a little bit on some of that research here. Really what this comes down to, and this is a bigger discussion, is the idea of, the value of breeding for organic, that there are genuine differences in terms of how organic systems work and how that might play into wanting to have varieties that are adapted to organic systems. Jim and Edith Landers Van Buren had written a review of some of those differences here, including things like mineral nutrition, crop rotation, weed management, tillage, crop protection, biodiversity, and seed treatments. All of these things we can see. Again, there's not necessarily organic may not be a monolithic system, just like conventional is not a monolithic system, but we can see differences, and those differences matter. And looking at how those differences play out, those differences can play out in terms of the performance of varieties, how varieties perform in conventional versus organic. There's been a lot of studies over the years that have compared conventional and organic and how varieties rank within those different systems. My research was not that. I'm standing on the back of other people that have made these comparisons and found in general that there are differences, and that those differences mean that if you are developing varieties using conventional breeding in conventional systems and moving them over to organic, that will impact the ultimate yield and resiliency and performance of those varieties, and that organic farmers are going to suffer because of that. So ultimately, if adaptation to organic systems is desired and there are changes in terms of the performance of how varieties do in conventional versus organic, then selection should be made in organic systems. And so this is a little bit of my project here, was saying, okay, well, if we're going to be doing that, we have these wonderful public institutions and public plant breeding programs like Dr. Tracy's, like Phil Simon, Carrot Breeder, who are working in these outcrossing crops, and a lot of the material that they have is in the recent in recent history, the bulk of that breeding, those breeding efforts have been in conventional systems. And how do we kind of access all of this elite material and find out what can perform well in organics and get it into organic systems? And so I'm going to get a little bit into the weeds of this specific project, but kind of the big picture was trying to have a case study where we tried to think of what are efficient ways to look through these breeding programs, find out what can do well in organics, and actually then turn around and produce varieties out of that. So I did a couple of things, taking advantage of the fact that we have both traditional mating designs, traditional plant breeding techniques for making predictions about the performance of how different parents might perform when they're combined together, as well as the advantage that now we have, it's cheaper and easier and more efficient than ever to start looking at the genomic sequence of these crops, of these varieties, and can we leverage some of that information to be able to do things more rapidly, more easily, or is there any value in that? So that was part of what I was looking at. In addition, I was also looking at some of these questions around, are organic systems monolithic for when we're trying to breed for them? How much do we need to be breeding for specific systems with organic? Are there traits that can be selected in one place that their performance will carry across lots of different organic systems? Are there traits where you really need to be breeding in specific regions? So those were some of the pieces of what I was looking at. Where we started with this was, and to maybe take a step back, it started with the idea that Dr. Tracy has all of this elite material, and a lot of his most elite material is in the form of these inbred parent lines. And so going through, we started with about 40 of his best inbred parents. And what we did was we then took those 40 parents and made deliberate crosses to make 100 hybrids. And a system that's been around for quite a while called a North Carolina Design Tube, developed at North Carolina State University as a mating system. And I'll show you what that looks like. There are these little blocks like this where you would take a set of 5 of the inbred parents here and another separate set of 5 inbred parents there and make all these different crosses between them. And then we did this basically 4 times to make the 100 hybrids. Now, you can start to see quickly there's lots of hybrids that we didn't make, and that's what I'm going to get to is, okay, of the hybrids we didn't make, and of all the other possible permutations as we extend this out to trying to make larger open pollinated populations, can we start with the smaller set of these crosses and start making predictions about how they might perform, how the untested crosses and the untested open pollinated populations might perform. So we made those crosses, and then what we did was we conducted these training trials. So in 2015 and 2016, grew the 100 hybrids and the 40 inbred parents in 6 locations. 2 sites in Wisconsin, 3 sites in California, and then up here in Oregon, all organically managed, all organically certified operations, but lots of different environments, from dry farmed to irrigated to rain fed, in different climates and in different parts of the country. Grew that both in 2015 and 2016, and grew them in, one of the other things was trying to come up with a good experimental design, again trying to be as efficient as possible because this can get overwhelming. How can you actually get good information back without having to go to, once we're starting to talk about 140 different entries, how many times can we really replicate that? So one of the things we did was we cut down to 2 replications, but then within each of those replications, we divided those into these smaller blocks that were interspersed with check varieties to start controlling for possible differences in the field within each of those larger replications. We measured throughout the year plant height, ear size, tip fill, husk cover, flavor, maturity, all traits that we knew were going to be relevant in the ultimate decision around what varieties would be good and not good. How we did quality was primarily all in a subset of locations, because quality in sweet corn is a challenge because the timing of when you taste and when you evaluate quality is it's a tight window. And so because of that, it was only a subset of locations. There's usually 3 of the locations each year. We would do a quality evaluation by bite testing, but then to validate that, I would also collect samples and freeze those to do wet lab analysis and also make predictions using this NIR prediction tool. So what did we get out of those first 2 years of these training trials, just testing all of these different hybrids and inbreds? We found that there were differences between the entries for all the traits. One of the things that we found was that there wasn't a rank change genotype by environment interaction, meaning that there were differences between how the genotypes performed at different locations. But overall, if something did the best for flavor in one location, that carried across the other locations. And that was true for all of the traits that we measured, which was kind of, frankly, a surprise to me, because I expected, I expected flavor, for example, to be relatively consistent, but some of the other, more agronomic traits like plant height or ear width, I expected to see more rank changes, and we weren't seeing that as much, which was possibly promising in terms of the idea of how far can you take something and carry it forward and test it in new regions. Again, this was only these 6 locations. There's lots of other parts of the country with, specific diseases that might impact some of these, and of course, there's lots of other crops where this wouldn't necessarily apply. We also saw differences in what we call general combining ability for all these traits. Let me tell you what general combining ability is. General combining ability is kind of a traditional way that we can look at the performance of an inbred as a parent, is how well do the hybrids that are made out of it perform. And so once we had all that data, we could go forward and start making predictions. And starting from the general combining ability of an inbred and say, okay, if we know that this is for flavor, for example, if this inbred, overall performs, in this case it's color-coded, green is tastier, and if it performs above average and another inbred here performs above average, the combination of those two, it's an additive model here, we expect to perform above average. So this is allowing us to kind of move out from the tested to the untested using this traditional design. But again, we also have the advantage of being able to genotype all of this. And then what that allowed us to do was make a similar set of predictions using genomic data, using high density sequence data to make similar predictions. And just briefly, we found that those predictions were fairly accurate sort of within our initial prediction trials. We would take a subset of the entries and say, how well do they predict the things that we already tested? And we found that the predictions were fairly good. But what we wanted to do is actually do outside of what we had been testing during those the initial trials. We wanted to do a new set of trials with new material to see how well now, when we carry it forward to new material, how well do those predictions hold up. So the following year, grew in 5 locations, a set of 24 of the hybrid entries, as well as some new open pollinated lines to test and see how well do they line up with what we predicted we thought how well they would perform. So in this case, likewise, we saw differences between the entries, and we didn't see any rank change genotype by environment interaction. The traits seemed to be relatively stable across regions. And what we found was that we had fairly strong correlations here between the predicted and actual values, both based on this traditional general combining ability based prediction, and also we saw improvements around using the genomic based, the genetic sequence based predictions. And some of the best improvements that we saw were around some of these traits that were controlled more by have more of a heterotic influence, where there may not be a simple effect where you can just add the 2 parents together and make a prediction, but there are possibly something in the combination of those parents that is synergistic. And the model that we used using the genomic data allowed us to have a little better read on some of those possible synergies between the parents. And so that's maybe why we see kind of this jump for some of these traits when we're to use the genomic data. But for many of the other traits, like flavor, using the traditional general combining ability based prediction was quite reasonable. And then for some traits, you can see in general our correlations are good, but here in tip-fill, we really were not seeing good predictions on either case. So just some quick little slides showing what that looks like between the predicted values and the measured values. For some of these different traits, like this is flavor. So, quick conclusions here. We saw that this method could be useful in terms of predicting, and that we did find that this idea of being able to look at dominance or heterosis could be useful for some of the traits. The other strengths that we had from this project is now we have this data set that, for example, if we're making these new open pollinated varieties using this information, and we can make choices to say, okay, if a farmer is really interested in something that's very high flavor but also has large ears, we can go back into the data set and make specific choices out of those different inbred parents to create new populations to start as breeding projects or possibly release as cultivars. And so we're also, some of those we are moving forward with seed company trials and have some promise in terms of commercialization. Thank you.
Great. Thank you. We have time for questions.
I wish I could form this question better than they come out of my mouth. But anyway, given the way that the reproductive biology of sweet corn, right, wind pollinated, likewise, how many, if I could use the word lines, how many lines are present when an open pollinated variety like the ones you're talking about are grown? I'm wondering about the year to year, within year, and then year to year variation.
Yeah. So for the when we were creating the training trials, one of the things that we did, one of the kind of convenient things about working with inbreds and hybrids is we were able to reduce some of that genetic variation in the training trials so that there was, genetic uniformity within each of those plots. As we move forward to some of those prediction trials where we were creating these new open pollinated varieties, then we see more variation. That being said, one of the things that we were able to predict, as well as the kind of mean performance of a variety for a specific trait, is we were actually able to make predictions around how much variation there might be in a given trait, so that we could look for some traits that may be less important in terms of the variation. Maybe overall plant height may be less important, where something like maturity is actually where the variation becomes more important than the mean. It's okay to say, okay, I've got something that's early or late as long as you can predict that, versus something that has a really wide range. So that was a tool. But maybe back to your point too, is that the results from the prediction of the open pollinated varieties were certainly fuzzier compared to the accuracy that we saw between our tests and between our predictions and our actuals for the hybrids, and a lot of that was because of genetic variation.
Jared, I was just wondering, from a sort of running a breeding program perspective, would you, ditch general combinability and use G-block models moving forward, or how do you balance those if they're both able to predict what you want? Yeah,
so I think what I would say, some lessons I learned there, one of the things is that we're working with corn, it's sweet corn, but it's still corn, and so we're able to leverage the fact that there's a huge infrastructure in place. We have, we have the complete genome. The prices for doing sequencing is, I think it was $40, for an inbred to get the sequence on it. So it's, cheap. That's not going to be the case as you move out with crops that are lesser studied and have less of that infrastructure. The other risk that I didn't mention, but is really important to keep in mind here, is a big risk that I see with the genomic prediction side of it is that unless you are intentionally including in your selection to maintain diversity, the models will push you towards uniformity and reduce the genetic diversity in your OPs, because the models will just say, okay, whenever you've got these specific, alleles, it, tends to be a little sweeter, it tends to be a little, taller, and so always, the models will always tell you, pick everything with those alleles, and so that will, even, more than when you have selection in the field, or even with the general combining ability, where there are more than one route maybe to get a sweet plant or a tall plant, and that genetic diversity can end up, even if you've got a phenotype, the thing that you're looking at may look similar, there might be different genetics underlying that, because with this genomic selection, you're looking at the genetics and making selections based on that. That carries a risk of reducing diversity unless you actively push back against that. Yeah.
Is the ultimate prize processing, fresh market, or dual use?
So for this, really, this was rolled into the Novik project, and we were looking at, we were looking at SEs and for direct seeding, and we were looking at, part of it was, creating this, hypothesis testing and building a model, but in terms of the kind of sort of stacked function of actually developing commercial things. My hope was to release some open pollinated varieties and populations, and so, because of that, because of the increased amount of variability that we would see in that, this is going to be, more of a fresh use, where a little more variation is going to be tolerated than, maybe the processing industry.
Great. Thank you again, Jared.
Yeah.